Hello, everyone, and welcome to our program today entitled Assessing Risks to Bees from Pesticides, the second in a two-part series of Advancing the Science of Pesticide Risk Assessment webinar. My name is Justin Hausinger. I'm a branch chief in the Office of Pesticide Programs. The format for today will be a series of 3 presentations on a variety of topics, and then we'll transition into a question and answer session with the panelists. This webinar is produced by the United States Environmental Protection Agency, and the views expressed by our presenters are for educational purposes only and do not represent the official views or positions of EPA or the presenters' organizations. EPA's Office of Pesticide Programs began hosting a series of 5 public webinars highlighting the ongoing work to promote pollinator health and habitat. This webinar is the 3rd of this 5-part series and second in a 2-part sub-series of Advancing the Science of Pollinator Risk Assessment. This webinar will focus on understanding and advancing the science of assessing risk to bees to support the registration or reevaluation of a pesticide and provides a brief overview of EPA's tiered process for assessing pesticide risk to bees. Since risk is a function of both exposure and toxicity, this webinar will include a discussion of both predictive and residue monitoring studies that EPA uses in higher tiered assessments to refine estimates and exposure models. The webinar will also provide an overview of honeybee colony simulation models and new approach methodologies that are under development to help reduce the reliance of in vivo tests. So we're pleased to have it with us a diverse and distinguished group of presenters today. I'd like to take this moment to introduce them. Dr. Thomas Steeger has over 40 years of experience in conducting and evaluating mammalian and ecological effects data. Tom currently serves as Senior Science Advisor in the Environmental Fate and Effects Division of EPA's Office of Pesticide Programs, where he conducts ecological risk assessments of conventional pesticides. Over the past 15 years, Dr. Steeger has worked with multiple stakeholders, both domestically and internationally, to develop a standardized process for evaluating the likelihood of adverse effects on bees from exposure to pesticides, along with the tests needed to inform that process. Keith Sappington has over 30 years of experience conducting applied environmental research in support of human health and ecological risk assessments. At the US EPA, Mr. Sappington also serves as a Senior Science Advisor in OPP, where he conducts ecological risk assessments of conventional pesticides. He currently co-chairs the semi-field, full field testing work group within the International Commission of Plant Pollinator Relationships, which is focused on developing technical guidance for higher tier testing with bees. Kristina Garber is also a Senior Science Advisor in the Environmental Fate and Effects Division. Chris has been working for several years to advance methods for assessing pesticide risk to endangered species and to bees. Ms. Garber has served as the lead in developing predictive models for estimating exposure of pesticides to both aquatic and terrestrial taxa, including the B-Rex model for assessing exposure and risk of pesticides to various castes of bees. Ms. Garber has also worked collaboratively with the U.S. Department of Agricultural Research Service in developing a pesticide module for honeybee colony simulation model, VivoPop. So now we'll transition to the first of the presentations today. Dr. Tom Steeger will speak to us about the framework for assessing pesticide risk to bees that will provide all of us with a springboard for the rest of the topics that we'll cover today. So Tom, you have the floor.
I want to thank you for this opportunity to speak. Today you're going to hear about our risk assessment process for bees, and to assess the extent to which pesticides can potentially affect non-target organisms, EPA requires multiple studies on a wide range of taxa, for example, mammals, birds, fish, aquatic and terrestrial plants, and aquatic terrestrial invertebrates. For aquatic and terrestrial animals, EPA requires acute and chronic toxicity tests. All of these testing requirements are specified in Title 40 of the Code of Federal Regulations, Part 158. The procedures used in these tests are recognized internationally as being fit for purpose. That is, they include measurement endpoints which are biologically relevant and are of regulatory interest. And the tests provide reproducible, relatively selective, that is, a reasonable low number of false positives, and sensitive results, that is, they're capable of detecting effects at environmentally relevant exposure levels. As indicated, the methods used in these tests have been suitably evaluated across a wide range of labs, that is, they've been ring tested. And the tests typically have curated reference chemicals with which to evaluate test performance. Risk is commonly defined as the likelihood and magnitude of an adverse effect from exposure to a particular stressor. That stressor could be a pesticide. Therefore, risk is a function of both the extent of exposure and the sensitivity of the organism to the stressor. And the sensitivity can vary depending on the taxon and species tested. Risk assessments can be deterministic, that is, based on point estimates, or they can be probabilistic and based on distributions of exposure and sensitivity estimates. Using a deterministic approach, EPA evaluates the ratio of the point estimates of exposure, typically the highest estimated environmental concentrations, to point estimates of toxicity, typically the lowest, most sensitive toxicity values, in what are referred to as risk quotients, and they're abbreviated in RQ. The agency then compares these risk quotients, or RQ values, to agency levels of concern, which represent thresholds for mitigation. As with many things, the agency's process for conducting risk assessments in support of regulatory decisions is described in a series of publicly accessible guidance documents. In 2011, EPA issued interim guidance on exposure and effects data on bees. This guidance was based on the agency's understanding of the evolving science through a 2011 Society of Environmental Toxicology and Chemistry Global Pelston Workshop on pollinator risk assessment, and by work underway in Europe through the European and Mediterranean Plant Protection Organization. In 2012, EPA, in collaboration with Health Canada's Pest Management Regulatory Agency and the California Department of Pesticide Regulation, presented a white paper to EPA's Federal Advisory Committee, the FIFRA Scientific Advisory Panel, describing a conceptual framework for addressing pesticide risks to bees. Based on that white paper, input from the Scientific Advisory Panel, and additional guidance documents generated through the European Food Safety Authority, EPA, Canada's Pest Management Regulatory Agency, and California Department of Pesticide Regulation released the harmonized guidance for assessing pesticide risk to bees in 2014. This guidance was considered by Environment Australia in their development of a roadmap for assessing risks of pesticides to bees, and the guidance has subsequently been translated into Spanish for consideration as a US, Mexico, and Canada harmonized guidance document. In addition to the 2014 guidance, in 2016, EPA issued guidance for agency risk assessors, which discusses the regulatory provisions for requiring data, the current pollinator data requirements for conventional pesticides at that time, and the additional bee toxicity and exposure studies. Effects testing includes laboratory-based acute and chronic toxicity studies of individual adult and larval bees, and field-based studies of whole colonies, as well as residue monitoring studies in pollen and nectar, which are used to refine estimates of exposure. For each of the acute and chronic toxicity studies of adult and larval bees, there are guidelines outlining the study protocols, which have been tested internationally to establish that they are reproducible and provide consistent results across multiple testing facilities. Many of these test guidelines are published by the Organization for Economic Cooperation and Development. It's abbreviated the OECD. Dr. Dan Schmiel and Dr. Ivo Rosnick discussed the efforts to develop such guidelines in the previous webinar. There are also study guidance documents for semi-field and full field colony level studies. While EPA has been evaluating risk to bees for over 20 years by requiring data on adult bees and when needed on whole bee colonies, the new data requirements address uncertainties regarding acute contact exposure to adult bees, but now include acute oral toxicity to adult bees, that's OECD Test Guideline 213, and acute toxicity to larval bees, OECD Test Guideline 237. The chronic toxicity tests with adult bees follow OECD Test Guideline 245, which examines young adult bees dosed for 10 consecutive days, while the chronic tests with larval bees, OECD Guidance Document 239, doses larvae for 4 consecutive days as they transition from the second instar until they enter pubation and stop feeding. These tests extend through adult emergence and therefore represent the full development of worker bees from egg to adult. In addition to monitoring for bee mortality, the acute and chronic toxicity tests must also report any sublethal effects that may occur during the study. Depending on the outcome of those tests, the risk manager's need for additional characterization of potential risks, additional semi-field OECD Guidance Document 75, and full field colony level studies, Office of Chemical Safety and Pollution Prevention Guideline 850-3040 may be required. In addition to the colony level tests, as I said, exposure data may also be required on residues in pollen and nectar. As I mentioned, risk is a function of exposure and toxicity. With respect to exposure, initial estimates are based on either default values or predictive models, which are part of the BREX tool developed by Chris Garber in the Office of Pesticide Programs. Chris Garber will be presenting today on some of our recent colony level modeling efforts. For contact exposure estimates from foliar applications, the underlying data used to develop initial estimates, referred to as Tier 1 exposure values, are based on measured residues in bees based on the published work by Kosh and Weiser following contact exposure. The exposure estimates for pesticide residues in diet for foliar applications are based on the Kanaga nomogram, which the EPA Office of Pesticide Programs also uses for estimating exposure to both mammals and birds. For soil treatments, exposure estimates are based on the Briggs equation, which considers chemical and physical characteristics of the pesticide relative to measured values in plant tissues. Similar to what is done in the European Union, a default exposure value of 1 milligram per kilogram is assumed for residues in diet from seed treatments. EPA is continuing to refine these predictive models as more data become available. As I indicated earlier, EPA may require residue monitoring studies in pollen and nectar. These data can be used to refine risk estimates that were initially based on predicted or default values. Keith Sappington will be talking about the residue monitoring studies in the next presentation. This is a simplified diagram of the 3-tiered process used by EPA to assess risk to bees, where the answers to multiple questions to determine the extent to which higher tier studies or refinements are needed. For example, the most basic question is whether it is likely that bees are exposed. This involves understanding whether the crop may be attractive to bees, whether applications occur at or near bloom, and the time at which the compounds are applied. Also, transitioning to higher levels of refinement is influenced by whether mitigation measures may be available and the nature of data with which risk estimates may be refined. As noted earlier, the process used to assess risk to bees is consistent with the process used with other taxa in that it is tiered. It consists of initial screening level, or Tier 1, in which conservative estimates of exposure and effects to individual organisms, for example, adult and larval bees, are used to quantify potential risks. The screen focuses on what are considered to be major routes of exposure, that is, contact and ingestion of residues in diet, such as pollen and nectar. Depending on whether or not the Tier 1 risk can be mitigated, the risk manager may indicate that additional refinements are needed, in which case higher tier assessments are conducted, where measured rather than modeled estimated exposure values may be used. Higher tier evaluations can also include colony level effects data. As with other taxa, and as noted earlier, the risk assessment process relies largely on guidelines, EPA and/or OECD guidelines, although one of the studies, residues in pollen and nectar, does not have a formal guideline or guidance document, and proposed study protocols must be reviewed in advance by EPA. Since preceding tiers are intended to represent refinements, Tier 1 focuses on individual bees, while Tiers 2 and 3 focus on colony level effects, transitioning from studies conducted under relatively controlled conditions, those such as semi-field tunnel studies or colony feeding studies, and culminating in full field pollinator tests in which typically full-size colonies with free foraging bees are studied under environmentally realistic exposure conditions. For the latter, the field-based studies, typically EPA requests draft protocols to review in advance of the conduct of the study so that we can understand the specific questions being tested and the methods being used to generate and analyze the data. While laboratory-based studies are typically conducted with technical-grade active ingredient, that is the pure pesticide, the colony-level studies are typically conducted with typical end use, or what we call formulated products. Therefore, our concerns regarding the effects of inerts, such as surfactants and adjuvants contained in the formulated product, can be evaluated in the colony-level studies. At Tier 3, though, all the factors influencing honeybee colonies, such as pests and diseases, pesticides, poor nutrition, bee management practices, and weather, all have the ability to impact the study. EPA is working with its regulatory counterparts and with researchers internationally to ensure that the development of exposure and effects tests that can be used to address uncertainties related to potential effects of pesticides on bees. Since the initial guidance documents were identified in 2011, additional acute oral and contact toxicity test methods with social non-apis bumblebees, which is a soil nesting species, have been developed in OECD Test Guideline 246 and 247. Dr. Rosenek spoke about efforts to develop non-apis toxicity testing protocols in the preceding webinar. The EPA Office of Research and Development has also participated in the ring testing that has been conducted to evaluate toxicity test designs for the non-apis solitary mason bee, which is a cavity nesting species, and has been involved with method development for chronic toxicity tests with bumblebee microcolonies. EPA Office of Pesticide Programs has also been engaged in a retrospective analysis of acute and chronic toxicity data on adult and larval bees that have been generated or submitted to EPA thus far to determine whether the full suite of toxicity tests is needed or whether certain tests appear to be protective. For example, honeybee acute oral toxicity tests appear to be if not more sensitive indicators of honeybee toxicity than acute contact toxicity tests. This information enables the agency to better direct toxicity testing and to reduce the number of organisms subjected to toxicity testing. As discussed during the previous webinar, EPA is continuing to rely on the honeybee as a surrogate for non-apis bees. In 2017, EPA hosted an international workshop facilitated by the Pollinator Partnership with researchers from academia, government, and industry examining exposure pathways for non-apis bees. The workshop indicated that while honeybees may be reasonable surrogates in terms of exposure through diet and via contact, there is uncertainty regarding the extent to which honeybees are protective for exposure of developing bees through contact with soil or with leaf materials. At that time, there were insufficient data to resolve those uncertainties. When data are available for non-apis bees, EPA considers these data on a case-by-case basis to determine the extent to which honeybees are not suitable surrogates. Data on non-apis bees will continue to be used qualitatively to characterize potential hazard to non-target organisms. EPA is continuing to be engaged in the development of guidelines for testing non-apis bees to ensure that suitable methods are available for testing these species and to provide more uniform data with which to evaluate the extent to which honeybees are suitable surrogates. As with studies of exposure to and effects on honeybees, EPA applies the same standards to evaluating the quality of these studies as those applied to other taxa. This presentation has provided a brief overview of EPA's process for evaluating risk of pesticides to bees. This process used by EPA to assess risk to bees has evolved stepwise with the agency's understanding of the science. The data used in the process represent internationally harmonized methods that reflect many years of review and collaboration. At this time, and on taxa assessed other than mammals, the process used to assess risk to bees is one of the most data intensive and provides the agency the means of assessing potential risk to bees from exposure to pesticides. Thank you very much for the opportunity to present.
Thanks very much, Tom, for giving us that foundational understanding of the risk assessment process for bees and the introduction on the various types of studies that comprise it. So Tom touched on the higher tier field testing studies, and we'll now hear from Keith Sappington, who will give us a closer look at one of those types of field studies, and those are the field residue studies with pollen and nectar. So Keith, I'll hand it over to you.
Thank you, Justin. Well, welcome everybody and good afternoon. And I'm really pleased to have this opportunity to share with you information on how EPA uses results from higher tier exposure testing in assessing the pesticide risk to bees. So last week's webinar focused on methods for assessing the effects of pesticides on bees. What I'm going to focus on are methods for assessing exposure, and particularly those that come from higher tier exposure studies, or what I also refer to as field residue studies. So for today's topics, I'll first describe the nature of higher tier exposure studies and their role in EPA's risk assessment process. And then I'm going to show some examples of how these data have been used in bee risk assessments. In addition, I'll provide some lessons learned that we gain from analysis of a large body of residue data for pollen and nectar. And then I'll summarize briefly a few efforts that EPA is supporting to help advance higher tier exposure methods. Before I continue, I want to briefly summarize the main features of higher tier exposure studies so we all have a common understanding as to what they really entail. And so broadly speaking, higher tier residue studies are experimental studies that are conducted in the field in which a pesticide is applied to a representative crop. And after that application is made, or multiple applications, samples are taken of bee relevant matrices such as pollen and nectar, and those samples are analyzed for the concentrations of the pesticide and any metabolites of concern that would be in those matrices. And these samples are usually collected at multiple points in time after the application. Now, these studies are usually conducted at maximum allowable application rates and minimum intervals in between applications to give us sort of an estimate of a high-end exposure. And importantly, they are replicated across multiple sites in different regions in the country where the crop is grown. And they may be conducted for multiple years. For example, if there's concern about pesticide carryover in soil from one year to the next. And so therefore, these studies are fairly resource intensive and often can take 1 to 2 or even 3 years to complete. So in order to show how higher tier exposure studies are used in bee risk assessment, this is a boiled down version of the risk assessment scheme that Dr. Steeger just presented. And we use higher tier residue data in 2 ways. And first is to refine our estimates of tier 1 risk, and recalling from Tom's presentation, that's risk at the individual level. And the second is to estimate exposure by which we would evaluate risk to entire colonies. So why are these studies important? First of all, they give us a chemical and crop-specific estimate of the magnitude of the pesticide of concern in the diet of bees. This is important, and I'll illustrate that with this plot here. On the vertical axis are the concentrations of various neonicotinoids in floral nectar of cotton, and on the horizontal axis are the days after the last application was made. And you'll see in the red box, this arrow is pointing to the default residue estimate from the B-Rex model. So at this application rate, which is 0.1 pounds per acre, we would estimate from the model 11,000 micrograms per kilogram. And clearly, in this case, the measured residue data, which are shown and coded by the chemical, by the different symbols, are at least 2 orders of magnitude or 100-fold below what we would estimate. So in this case, it's clear that having the residue data represents a major refinement of exposure for, in this case, residues in cotton nectar. Now, that's not always the case. we have many examples of where our measured residue data are very close to the predicted values. The other thing, and this is equally important, is we have information that can allow us to evaluate the duration of risk as well as the frequency at which that risk might occur. And so without these data, we essentially have a point estimate, and we really don't have a viable way of estimating duration and frequency of risk. So I'm going to transition into how these residue data are applied, and I'll first talk about Tier 1 portions of our risk assessment, and then I'll also provide an example of how they're applied in Tier 2. But first, I want to point out that currently EPA does not have formal guidance on exactly how to use these residue data in bee risk assessments. And I think that's in part due to the fact that when we developed our risk assessment guidance for bees back in 2014, most of the experience at that time with higher tier studies had to do with estimating effects on colonies rather than exposure. So we really didn't have the experience at that time to really develop such guidance. So what I'm going to present to you represents our current practice. And that practice has been based on our experience with the risk assessments for the neonicotinoid insecticides as well as several other pesticides. So at Tier 1, what we can do is use the full set of residue data in the BREX model that Tom described earlier. And we can develop acute and chronic risk quotients using all of the data. And this can be developed for different castes of bees or bees with different tasks, such as adult nectar forager and larval bees. And this plot shows the output from a BREX run using an entire data set of residue data. And on the y-axis is the oral risk quotient, or RQ, and on the x are the days after the last application. And the horizontal red lines represent our levels of concern at Tier 1. And the solid red line is the level of concern of 1, and that applies to chronic exposures. And the dashed red line is a level of concern of 0.4, and that's for acute exposures. And so what we can do here is evaluate the change in risk over time, and we can evaluate how the risk changes by the different life stages of bees as well as the different castes. So in this case, the adult chronic RQs shown in the solid blue circles are obviously quite a bit higher than those for larval bees as well as acute exposure to adult bees. So application of the residue data in Tier 2 is similar to that in Tier 1. I should mention that Tier 1 is all done on a dose basis. So the BREX model incorporates the amount of food that the bees would be expected to consume for the different castes and the different tasks. In the case of Tier 2, we use the residue data directly in comparison to our colony level toxicity thresholds. And these are derived from whole colony feeding studies where the food is spiked at different concentrations of the pesticide that would give us a dose response curve. And what we usually derive from these colony levels feeding studies are the NOAC, that's the highest concentration that does not cause a significant effect to the colony, and the LOAC, which is the lowest concentration that did have a significant effect on the colony. And so in this plot, again on the y-axis are the concentration of a pesticide in floral nectar. This is for cotton, and the x again is the time or the days after application. In this case, the solid red horizontal line represents the LOAC from the colony feeding study, and the dashed line represents the NOAC. So the dashed line is the highest concentration in the diet of the colony that did not cause a significant effect. And you can see early on we have exceedances represented by the orange triangles of both the NOAC and the LOAC. But later on, as time continues and the pesticide dissipates in the pollen, or in this case in the nectar, we see that those residue values drop below the colony NOAC. And we can characterize the strength of evidence in this risk, both from a temporal and a spatial perspective. So temporally, much like with tier 1, we can look at the magnitude, the duration, and the frequency by which these colony level endpoints are exceeded. And from a spatial perspective, we can look at the number of sites where we see exceedances. So is this occurring just at one site, or is it occurring at all of the sites for which we have data? Another thing that we've done with these data, you can see that there's gaps naturally in when the residue values are measured over time. And so we can use the measured residue data and use models such as regression modeling or other models based on the kinetics of the dissipation of the pesticides to sort of fill in these gaps in between when the pesticides were actually measured. And importantly, we can use these models to incorporate the variability that we see in the residues at any given time. And so this solid black line represents the 90th percentile estimate of residues and that decline curve over time. So 90% of the sites would have residue concentration below this. And the dashed line represents the median value, or the 50th percentile. And where these sort of curves can come into particularly useful manner is for estimating how long you might need before bloom in order for the residues to drop below, say, a colony level risk. So at the 90th percentile, for example, it looks to be about 16 days before bloom, you would estimate before the residues would drop below a colony level risk. So I'm going to transition now to some of the lessons learned from the neonicotinoid residue data. This database really is the largest database we have on residue data for a pesticide class, and it contains over 80 different residue studies that were submitted to the agency. And all of this information that I'm going to present is found in these documents, which are available online. There's 5 main points I'm going to summarize. And the first one is the effect of application rate on the residues in pollen and nectar. And as we would expect and certainly hope, the residues in pollen and nectar for the neonicotinoids generally scale by application rate within an application method. And to demonstrate this, the plot on the left shows residues measured in orange nectar of thiamethoxam after soil application. And in this case, the residues are measured or not normalized to application rate, and they're reflecting application rates that range from about 0.07 pounds to about 0.5 pounds, so almost an order of magnitude. And they're color coded as such, so you can see how they're sort of falling out in accordance with application rate. And the right plot shows when we have normalized all of these residue data to the same application rate. So clearly we've reduced a lot of the variability, but certainly not all of the variability. Now, with these particular data, the data points come from different sites. So not all of them are from the single site. So we're naturally going to have variability in the data due to other factors. But this is something that we use for other taxa, and we've seen in other plant tissues, but we felt we needed to demonstrate this in pollen and nectar, because once we normalize by application rate, we can then incorporate studies that have been conducted at different application rates and essentially expand our database for risk assessment. Now, the second observation I wish to point out has to do with the effect of the method of application. And by method, I'm referring to foliar spray versus soil versus seed treatment applications. And the application method, as you might expect, has a large impact on the residues that we see of the neonicotinoids in pollen and nectar. This plot on the left shows residues in various cucurbits for different neonicotinoids, so pumpkin, cucumber, and watermelon, after foliar spray applications. And you can see that residues start off about 5 days after application, and they approach 1,000 nanograms per gram. And these are all normalized to 0.1 pounds per acre. But there's an exponential decline over time in these residues. And at about 30 days, they're generally less than 10 nanograms per gram, and many are less than 1 nanogram per gram. So when we look at the same cucurbit crops, but we look at soil application of the neonicotinoids, two things are apparent. First of all, the overall magnitude of the residue is about an order of magnitude or more lower than what we see from foliar spray. And keep in mind, there are different scales here. But secondly, we do not see this declining trend over time that we see with the foliar spray applications. There's no obvious trend over time. Within some of the data sets, there are trends, but when you combine them, you do not see that. And we think the reason is that if you think about a soil application, you have mass of that pesticide that's remaining in soil, and that is available over time for uptake into the plant. So it's not a one-shot sort of application. There's a continuous exposure through the root zone and translocation to the pollen and nectar. For seed treatments, residues are generally very low in the few parts per billion range or a few nanogram per gram range, and they almost always were less than 10 parts per billion. Third observation has to do with application timing. We looked at pre-bloom versus post-bloom applications. And the plot on the left is a residue decline profile for various neonicotinoids applied to orchard crops. And you see again that characteristic exponential decline over time. And the residues are approaching about 3,000 nanogram per gram soon after application. For post-bloom applications, so these are perennial crops, and so post-bloom applications are obviously done after petal fall, but then the residues are measured during the next growing season when the crop reblooms. See that most of the residues are 10 parts per billion or 10 nanograms per gram or lower. And obviously the post-bloom application, there's a longer amount of time for the pesticide to dissipate. So that's partly, we think, the reason why we see lower residues. But it could also be a function of other processes such as leaf drop and losing residues through other means. Some of the largest differences we've seen, the residues in pollen and nectar for the neonics, has to do with the effect of site and crop. Typically, we see a 10-fold difference across sites and sometimes 2 orders of magnitude. So this plot on the left shows concentrations of clothianidin and extrafloral nectar of cotton, a function of the time after the last application. And the different symbols represent different sites at which the pesticide was measured. And so to get a sense of the effect of site on the residue measurements, you can compare these symbols or these residue values at similar points in time. And so near 14 days and about 21 days after application, we see that the total range here is about 100-fold. And so site can have a very significant difference. With respect to the effect of crop, this plot here shows thiamethoxam in nectar of various cucurbit crops, all measured at one site in Missouri. So this is squash, pumpkin, and melon that are represented by the different symbols. And when you compare residues at similar points in time, they're actually pretty close. Most of them are within a factor of 2 or so. But then with this same study, if we were to look at a different site in California, we get a very different picture. Here, the squash is clearly amongst the lower residues, while residues from melon and pumpkin are about an order of magnitude higher. So the effect of crop can be expressed differently, I think, depending on the site, and that could affect differences in the agronomy or differences in the hydrology or weather conditions and so forth. The final point has to do with the effect of the chemical itself on the residues. And so we looked where we had the same crop, we looked at how the residues compared among the neonicotinoids. And so this is an example of such data from melon following soil applications, and these are residues in nectar. And the different symbols here represent the different neonicotinoids, so thiamethoxam, imidacloprid, and clothianidin. And what we observed here is that the residues at a similar point in time after application generally vary by up to an order of magnitude. And I should point out here that we didn't have the situation where we had the 3 neonics, in this case, applied at the same site. So the variability you're seeing here can in large part be described by that, what I showed previously for different sites. So with the same chemical, we see an order of magnitude commonly, if not more, on the effective site. So with different neonicotinoids, we do not see a clear signature within the context of the site-to-site variability. So lastly, I'll wrap up by touching on several efforts that we're helping to support to advance residue study methods and application. And one is being led by the ICPPR, that's International Commission on Plant Pollinator Relationships. And there's a work group of this organization that's focused on developing more detailed guidance for how to conduct residue studies. And that guidance will be customized based on the actual purpose of that study. Residue studies can be done for different objectives. And experts from around the world who have conducted these studies or who have been reviewing these studies have been convening to develop this guidance. Another effort that we're helping to support is the development of a database that would contain the pollen and nectar residues as well as residues in other plant tissues. And from this database, we hope to refine some of the default values that Tom mentioned earlier, and I also presented from the VREX model. This database is also useful for asking questions in terms of the design of a residue study, such as how many sites or how many crops, similar to the data I just showed. But it will be much broader than just one class of pesticide. Finally, EPA is considering the development of additional guidance for exactly how to use the residue data in risk assessment. This will likely be a longer-term effort, and I think would require curating the residue data and conducting the necessary analyses so that we can use an empirically or data-driven approach to help us codify our approach for using residue data in risk assessment. And so with that, I will finish my talk, and I really thank you for your attention. Here are some links to some documents which I cited earlier in the assessment. So thank you very much.
Thanks for that, Keith. And I think all the visual tools that Keith was showing, just really hits home one of the central points of just how many factors and considerations go into these studies and just the enormous amounts of data that they produce. Thus far, there's been a discussion more of the deterministic methods where the exposure and effects data are derived from. Ms. Kristina Garber now will discuss kind of an emerging frontier, and that is some of the predictive tools for assessing pesticide risk to bees. So, Chris, you have the floor.
All right, thanks. So for the remainder of the webinar, I'm going to discuss some new methods that are currently in development for predicting effects of pesticides on bees. These methods aren't part of the standard risk assessment methodology that Tom described earlier, but they may be relevant in the future for risk assessment purposes. So I'll discuss 2 projects, and they're at 2 completely different levels of biological evaluations. The first, which I'll discuss in more detail, is a honeybee colony simulation model that essentially translates effects that occur to individual level bees and then translates those effects up to the colony level. The second model that I'll talk about is CECAPAS, which is at the protein level. So what it does is looks at similarity of protein structures to determine whether different species will have similar susceptibility to a given chemical. So starting out with the colony simulation model, as Tom described, the risk assessment method for bees that we use in EPA relies on a tiered process, starting with individual level effects data, which is tier 1. If additional information are needed, colony level data might be called in as tier 2 or tier 3 effect studies. And those are typically done in a field or in a controlled environment, a kind of semi-field environment. Now, a colony simulation model might be useful in determining whether tier 1 data might suggest a colony level effect independent of those empirical studies. So this type of model can incorporate bee biology, environmental variables such as weather and other stressors that we know are relevant to colonies, so that we can interpret the effects that happen at the individual level to the colony. Now, a colony model might also be helpful in identifying key elements that we might want to incorporate into a higher tier study design. So not all higher tier studies need to be designed in the exact same way. They should rely upon the available information for a chemical, such as the tier 1 data. And so this might also be an additional tool that could be used in helping design or even interpreting those higher tier studies. Several years ago, EPA presented a risk assessment methodology to our science advisory panel. This is something Tom talked about earlier. And in that, we identified kind of our wish list, if you will, for a colony simulation model. We want a model that can account for major factors that influence the dynamics of a colony, both individual colonies, so looking at the kind of complicated social structure of a hive, as well as variability from one hive to another. So, we know that the colonies can have very different responses to stressors depending upon their characteristics. So we want to be able to account for that. We want to be able to account for measurement endpoints that are directly relevant to our assessment endpoints and our protection goals. So having an idea of the strength of a colony, which is essentially how many bees there are, how many foragers there are, the likelihood that a colony will survive, as well as production of hive products such as honey, are desired features. We want a model that we can parameterize using data that we have available, whether it's the biology of the bees or the pesticide itself. A model that requires data that aren't typically going to be available to us, especially for specific pesticides, isn't going to be particularly useful. We want a model that is mobile, essentially, across the country. Office of Pesticide Programs assesses national level uses of pesticides. And so we need to be able to account for different areas of the country. So hives might be in California for almond production, for example, early in the year, and get exposed to a pesticide at that time of year and in that location. And then there might also be hives later on in the summer out on the East Coast for apples or blueberries or what have you. So we want to be able to account for differences in timing and differences in location that could occur depending upon the crop or the location. Of course, we want a model that is scientifically based and transparent to the public. So we want something that's documented and clearly defined, and some kind of standard modeling practices would involve the availability of a sensitivity or an uncertainty analysis, as well as an evaluation of the model and its predictions compared to empirical data, which is sometimes called a validation, but we typically use the term evaluation. So when we met with the SAP, really there wasn't a model that we could pick up off the shelf for pesticide exposure that really could address all of these desired features. So we essentially started the process of developing a model that could meet those needs for a pesticide application. And looking around at the available models, we settled on the BPOP, later called the Varroa POP model, which was developed by the USDA. Gloria DeGrande-Hoffman is the lead author on that. And this model addresses a lot of the desired features that we were interested in. So it includes colony dynamics. The model outputs that are generated are well aligned with the measurement endpoints that we would typically use in our risk assessment framework. Importantly, it also accounts for a number of non-pesticide stressors that are relevant to the impact of the dynamics of colonies, including varroa mite infestations and weather. The model's based on empirical data that evaluates different relationships that occur within the hive between different types of bees. The model has been published several times as it's evolved through the years, and that's included some sensitivity analyses and model evaluations comparing the model outputs to field data. So a little bit more about just BPOP, the original version of the model. This model essentially updates the colony population, which is essentially the number of bees within the hive, depending upon how many bees it started out with, the weather that is relevant to that hive, namely whether or not it rains, and the temperature as well. So that influences whether bees are out foraging. Queen strength is essentially how many eggs are being laid by the queen, and the more eggs she lays in a given day, the stronger that queen is. And then also the length of the lifespan of a forager bee. The sensitivity analysis that was conducted on the original bee pop model indicated that the queen's strength and worker longevity essentially are two of the most important parameters. Weather can also have an influence depending upon the timing of the year. In 2004, USDA published the Varroa pop version of the model. Essentially, this was a module that was added into the bee pop model to account for the dynamics of Varroa infestation in a given hive. Essentially, what it does is it tracks the number of Varroa mites that are present in a hive depending upon the infestation rate of the brood, and it also accounts for how many mites are present on the adult bees. Depending upon what the infestation rate is, it'll adjust the lifespan of worker bees, which, as I just mentioned, is an important parameter for the model. So there's kind of a feedback here where if there's a certain infestation rate of Varroa, then there'll be a decrease in the lifespan of bees and an impact to the colony. One really interesting aspect of the Varroa pop model is it allows for the ability to treat the colony with a miticide and essentially simulate the effectiveness of the control of Varroa and then the positive impact on the colony. And you can actually look at timing of that miticide treatment and the effectiveness of timing. So getting to the current effort. So the BPOP model and the Varroa pop model are both existing models that have been published before. And the current effort that I'm talking about is essentially adding another module into the Varroa pop model that accounts for pesticide effects. This is a collaboration between USDA and then EPA, and the EPA team includes members of both the Office of Pesticide Programs and our Office of Research and Development. This pesticide module essentially accounts for incoming pollen and nectar that are contaminated with a pesticide that's being assessed, and it accounts for essentially the kind of nectar and pollen requirements of a colony on any given day, and then those resources are either consumed or they're stored depending upon essentially how many mouths there are to feed and how much they need to eat. And that's all tied to the BREX intake rates that Tom and Keith both talked about. The model also accounts for contamination levels that are in the food that is stored. So if there's excess food, it'll be stored in the honey and pollen that's in the hive. And so we can essentially track kind of a delayed exposure that might occur. And then the model accounts for mortality depending upon what dose is received by larvae or adult bees and how that might correspond to the toxicity information that we have available. The exposure methodology has two different approaches. We can run the model using the kind of BREX default exposure assumptions for foliar, soil, or seed treatment, and then that essentially uses the same concentrations on pollen and nectar that are incorporated in BREX. Or we can use empirical residue data, such as what Keith described, and that would be the exposure concentrations that colonies would be exposed to. The effects data that the model uses are essentially the tier 1 data that would be part of the standard suite of studies for adults and larvae from laboratory studies. We also have the ability to count for some sublethal endpoints if we wanted to. So, for example, if there's a decrease in foraging, we could change the foraging rate of bees. The model accepts weather files that are also used by our PRISM model, which is our aquatic exposure model. And that has weather files from across the country. So essentially we can move the hives around the country and select a weather file that is representative of the area that we want to simulate. The Varroa pop model and the bee pop model also simulate one colony at a time, and we wanted to be able to account for variability from one hive to another. And so we've developed what we call a wrapper that allows for a Monte Carlo simulation of multiple hives. So we can account for differences in queen strength and worker longevity and other parameters as well, and how hives of different strengths might respond to pesticides. So the outputs of this model are very similar to what the original BPOP and Varroa pop model generate. So we get numbers of adults and number of brood, which is broken down into eggs, larvae, and pupae. And we can also basically tabulate how much mortality occurs at those different life stages as well. New to the model is an estimate of how much pollen and nectar is generated. And all of that is compared to what we would call as like a control or maybe a reference scenario is better, where we would simulate the hives without a pesticide exposure. And then we also run a simulation with the pesticide exposure and essentially compare the results. Let me show you what that looks like. This is just an example. The top row of figures is the number of foragers over time, and each figure is approximately almost a month's worth of time. The blue dotted line at the top of each of the figures is the 90th percentile estimate of number of foragers in the hive. And then the black line is the median estimate. And then the red dotted line is the 10th percentile. And so this is a summary of 1,000 different hives and their responses. So in the control scenario, you can see kind of over time, the number of forager bees that are present in the hives across 1,000 hives that are simulated. And if you look at the bottom row, that's dimethoate results. Now, dimethoate was chosen because that's a typical positive control for bees, as many in the audience, I'm sure, are aware. Essentially, the first two blocks or figures are before the application occurred, and you can see this blue arrow that represents when the application occurred. And then you start to see this difference, where you start to see the median dip down relative to the control or the reference, and you see that prolonged decrease. So not all hives were necessarily impacted, but if you look at your stats here, the median towards the end of the year, you can see the hives are still impacted. They have a lower number of foragers compared to the control. Let's switch gears a little bit and talk about CECAPAS. So as I said, this is at the other end of the level of biological organization. So CECAPAS is a tool that's been developed by our Office of Research and Development. And essentially, if you look on the EPA website for CECAPAS, you can find a lot of great details about this tool and the tool itself as well. And there are a number of publications available as well for CECAPAS. So this is essentially an online screening tool that allows for extrapolation of toxicity information across species. For some species like honeybees, we have a lot of data, but then for other species like a lot of our native bees or even a lot of species of bumblebees, we don't necessarily have toxicity information. So this is a potential tool where we could look at protein structures that are relevant to the mode of action of a given pesticide. And, we could look to see, okay, well, do other species also have that protein or that chain of amino acids? And essentially, it can allow us to look at that at the molecular level, whether we might suspect there's a similarity or a difference in susceptibility. So this model relies upon publicly available information on amino acid structures for different species and protein structures, might even have tertiary structure data available. There are a couple of publications that are available on this tool that have specifically looked at pesticides and looking at cross-species susceptibility, specifically for pollinators. There's a publication that was put out in 2016, looked at some insect growth regulators, methoxyfenozide and tebufenozide, that are believed to really target lepidopteran pests of crops. So some comparisons were made between a specific species of lepidopteran and other species of insects to see if that effect or other species such as bees might be susceptible. And so kind of what was found in that case study was that a number of species of lepidopterans maintain that protein structure of the target of those chemicals. There's another case study that was in that same publication that was looking at neonicotinoids and looking at the nicotinic acetylcholine receptor to see if that particular protein structure was conserved across different species of insects as well. And those comparisons were made between a honeybee and other species of insects. And essentially, there were similarities observed between those target proteins between bees and a number of different insect species as well. That is the conclusion of my portion of the talk. Just want to thank you for allowing us the time to talk about some of the work that we're doing.
Thanks very much, Chris. And I echo those sentiments. I want to thank you as well as Dr. Steeger and Mr. Sappington for taking the time with us today and sharing some of your knowledge and experiences in these important areas. We really appreciate it. I'll start off with the first question. How does one quantify a significant effect on a colony? Dr. Steeger, do you want to take that one?
Let me just say that the levels of concern that the agency uses to gauge risk quotients were developed based on the level of impact. So the acute risk level of concern for individual bees is 0.4, and for chronic risk, it's 1.0. The method of development and the levels of concern are discussed in the 2014 guidance document. The agency did not develop levels of concern at the whole colony level, yet we do qualitatively characterize effects on the colony. And these effects that we look at are on not just involving frank mortality of individual bees, like worker bees or brood, developing bees, but try to look at the whole colony and whether the effect is transient, that there might be an initial 24-hour mortality in response to a pesticide application, but to then see whether the colony, in terms of developing young, food reserves, adult bee numbers, return to control levels within a short period of time. And is the reproductive cycle of the colony every 21 days, the larvae are coming into developing as adults, and that's what's called a brood cycle. And we're interested in understanding other cases where the brood cycle is in any way impacted. And for the longer term studies, we answer questions as to whether overwintering capacity of the colony is in any way adversely affected. All of these effects are relative to the controls, and we're considering the extent to which these effects that are observed at the colony level relate to effects that are observed on individual bees in laboratory-based studies. So while we don't have a level of concern at this time for whole colonies, we're looking at the weight of evidence and how the overall colony is performing relative to exposure.
Thank you, Dr. Steeger. So for our next question, what is the criteria to identify a representative crop, assuming that the pesticide has crop grown approved on the label? Mr. Sappington, do you want to take this one?
Sure, it's a good question. And the short answer is going to depend. And it's going to depend on the attractiveness of the crop. It's going to depend on the risk profile at tier 1. And it's going to depend on how much the pesticide is being used on the crop. And in general, once we meet those criteria, if it has significant uses, and currently we like to see at least one crop within that crop group to be represented. And that's within the application methods. So if you have different methods of application, such as soil versus foliar, then we would be looking at different studies.
Great, thanks very much, Mr. Sappington. So the next question, can this modeling look at the cumulative exposure over the life of a colony to account for the movement of colonies over the year to environments where they may be exposed multiple times to some pesticides or to combinations of pesticides. I'm assuming that refers to the varroa pot model. So Ms. Garber, can I ask you to take that one, please?
Sure, I'd be happy to. Yes, we can simulate applications that might occur at different times. And we have actually been talking about simulating hives that are moving for pollination services. So almonds to apples to wherever else they might go next in the different environments and chemical exposures that might occur there. So that is a possibility that we're currently investigating.
Thank you. The next question we have is, what is the timeline for efforts that are underway at the EPA to create the residue database and formal guidance on how to use exposure data in the risk assessment process? Since exposure and toxicity have equivalent importance for risk assessment, this enhanced consideration of exposure is very welcome. Mr. Sappington, I'll turn it over to you for that one.
We are just now beginning to pull the data together. There is a database that the European Food Safety Authority has developed on residues in pollen and nectar, and we're using that information, and that is publicly available, and then incorporating information from other pesticides. I think that database, we're looking at probably 6 months to a year before that is up and available. During that time frame, we have to format the information and get it QA'd and so forth. In terms of the residue guidance from the ICPPR, we're looking to have that roughly within a year from now. There is a meeting being planned next year, an international meeting of this group, and we very much want to have that to be able to share with folks at that time.
Thanks, Mr. Sappington. So the next question I have here is, how does EPA estimate pesticide exposure to bees without knowing how much or how often a pesticide may be used in the future? Ms. Garber, I think this relates to the modeling efforts, so I'll throw it over to you.
I'll just say it can relate to the colony model, but this could also apply to how we do our standard risk assessment methodology as well. So generally what we assume is that the bees are consuming 100% of their food resources from the treated area. And so for running bee recs, we would use the exposure estimates for pollen and nectar from the treated crop. Same thing with residue data that Keith described. Now, if we have risk concerns, either at the single individual level or the colony level, then we can actually look at the influence of that assumption that 100% of their food's coming from the field on our risk conclusion. So we can basically kind of back off and say, well, what percentage of their diet from that treated area does it take to kind of exceed a toxicity threshold, either for individual or colony level? And then it becomes kind of a qualitative argument. So essentially, the lower that number is, the lower that percentage is that would trigger a risk concern, the more likely it is that they can get exposed to that, because we know that they collect resources from a diversity of places. So if that percentage is a small number, then the likelihood they're going to collect a portion of their food from that treated area is greater. So essentially that becomes kind of a qualitative discussion that can be considered in the risk assessment.
Thank you, Ms. Garber. The next question is, does the ICPPR still exist? Their website lists 2014 council members under the What's New heading. Dr. Steeger, can you please take this one?
Yes, the International Commission for Plant Pollinator Relationships, more specifically the Bee Protection Group, which Keith and I are both participate in, still exists. It had its international symposium last year in Bern, Switzerland, and it will have its next meeting in the UK, as Keith discussed. This is a very active group. It has multiple work groups, a microbial work group, the non-apis bee work group, exposure work group, field testing work groups, all collaborations between academia, industry, and government at an international level to help inform OECD efforts to develop guidelines and guidance documents.
Thank you, Dr. Steeger. The next question is, Mr. Sappington's presentation, the slide showing variation among sites in the amount of pesticide moving into pollen and nectar, was this for the same formulated pesticide, that is, holding the formulation constant? Mr. Sappington, I'll throw that one to you.
Yes, the slide that I showed that showed differences across site, that was within the same study. That was a study done by California Department of Pesticide Regulation. And I believe in that study, they looked at 9 different sites. And so that was the same, product. It was the same study.
Thanks very much. The next question is, what is the timeline for the Varroa Pop plus pesticide model? When will it be publicly available? Ms. Garber?
We don't have a date at this time for when the model will be made available. We're still in the evaluation phase. We do have a publication that we're currently working on that would describe the evaluation against empirical colony data, but at this time, the end date of the release is not determined.
The next question, what work has the EPA done to evaluate effects of pesticides on our native bees, and taking into account that there are more than 4,000 species in the United States and most are solitary and nest in soil, different to what is described on honeybees? Dr. Steeger?
EPA considers the honeybee as a surrogate for non-apis bees, both social and solitary. And the agency recognizes, as with other taxa, that there are multiple hundreds, thousands of species that the surrogate is intended to be representative for. With that said, the agency has focused on ensuring that we have a full suite of tests for evaluating effects on individual bees and on colonies, again, serving as surrogate for social and solitary apis and non-apis bees. But the agency has worked with researchers in academia, government, and industry on ensuring that there are suitable test methods for other non-apis bees. The test methods have been developed for bumblebees for acute and chronic toxicity for acute effects. Those are test guidelines now under OECD. There is a microcolony test. The EPA Office of Research and Development actually helped develop methods that are used to evaluate bumblebee microcolonies. And as Dr. Ivo Rosnick talked about, there are efforts underway to develop acute and chronic toxicity tests for solitary non-apis bees, specifically the mason bee, Osmia lignaria. Methods are also being developed for alfalfa leafcutter bees, Megachile, and the agency recognizes that you can't test everything, but we do try to have reasonable battery of tests to ensure that we understand the potential effects of pesticides on different taxa, and where there are uncertainties regarding the potential effects on non-apis bees, and when there are data on those bees, the agency considers those data on a case-by-case basis.
Thank you, Dr. Steeger. I think we have time for one more question. So Mr. Sappington mentioned that measured residues in cotton tend to be much lower than those predicted by BREX, while other measured residues were similar to those predicted by BREX. Is there a trend in which crops tend to have measured residues similar to those predicted by BREX and which tend to have lower residues than those measured by BREX?
I want to make sure that folks understand that the data I presented was for one class of pesticide. And in fact, the data I presented was for floral nectar. We also had in that same study extra floral nectar, and in that case, the residues were about within an order of magnitude of the BREX estimate. So, it's not just even a crop-specific situation, but here we have differences in which portion of the plant residues are expressed in and how that compares to BREX. The short answer is no, we've not done that comparison, and I think in part because we need to pull the data together for much more different chemistries in order to see if there is a crop-specific bias. It would be great if we could identify a worst-case crop, if you will, in terms of residue levels, I think that'll be difficult given what we know about the differences across sites. But we'll be able to address that question hopefully in a year or so when we have a lot of this data pulled together.
Thanks, Mr. Sappington. So we're out of time for questions. On behalf of EPA's Office of Pesticide Program, I again like to thank our panelists today and for all of you for joining us. Have a great day.